We have seen features of tight junctions. Now let's look at some of the molecules that make the functioning of these junctions possible. So let's look at those. Claudins are the major transmembrane adhesion proteins in a tight junction. They are essential for tight junction formation, function, and claudins in different claudins in different types of tight junctions. We talked about two different types of tight junctions. One, they're in epithelia, for example, in the gut, and the others that are present in urinary bladder, for example. A sealing strand has a long row of claudins embedded in both interacting plasma membranes. So here we have plasma membrane of one cell. This is a lipid bilayer, and this is the lipid bilayer of the other cell, and this is the cytoplasm of cell number 2. This is the cytoplasm of cell number 1. So here we have claudins. They are present in opposing plasma membranes, and two claudins, they come together and form these sealing strands. We saw their structure under the electron micrograph in the previous module. There are also another type of protein which are present in tight junctions called occludin. These occludin proteins, shown here in light green, the function of these proteins is sort of uncertain at the moment. However, they are thought to play some role in formation of tight junctions. A sealing strand has a row of claudins embedded in both the interacting plasma membranes. Extracellular domain of claudins join directly to one another to occlude the intracellular space. So this interaction is going to prevent any material from getting through. So the space here is very, very tight. As we have discussed earlier in our previous course, proteins carry information in two ways. Number one is that structure or the chemistry of the protein on its surface, and number two, the shape. So we talked about how two proteins can interact. It is dependent upon the shape, three-dimensional shape of the protein, and the moieties which are present on that surface. So here the structures of this protein is such that the two proteins can bind each other very, very tightly, and they prevent any flow of any material through the space in between them. A specific claudin found in kidney epithelia is required for magnesium to be reabsorbed. The magnesium is reabsorbed from urine back into the blood, and this type of claudin is specifically found in kidney epithelia and not other tissues. A mutation in the gene encoding this type of claudin results in excessive loss of magnesium in urine. The ability of tight junctions to restrict the passage of ions through spaces between cells increases logarithmically as the number of these strands in the network increases. Basically, what this means, or this information is suggesting, that each strand acts as an independent barrier to the flow of ions. Claudins and occludins are associated with intracellular proteins which are present in the cytoplasm, and these proteins are called ZO proteins. ZO basically means zona occludens, which anchor these ZO proteins, anchor the strands of actin cytoskeleton to the claudins, and thereby enhancing their function. As I mentioned earlier, tight junctions, claudins, they are not present in the invertebrates. They don't make these tight junctions. They make a different type of junction called septate junction. They are the main occluding junctions in invertebrates, and they form a continuous band around each epithelial cell. Mutation in zooproteins, zona occludens proteins, analogs in the invertebrates cause septate junctions, cause lack of septate junctions, and these animals, they develop epithelial tumors. This suggests that normal epithelial cell proliferation may depend upon intracellular signals coming from occluding junctions. So not only these proteins are sealing two membranes of two cells together, they're also providing signals to the cell, and they're interacting with the cytoskeletal element of the cell also. In the case of these tight junctions, the cytoskeletal element is actin filaments.